There is truly no scent that signals summer, or a celebration, or just, simple pleasure, quite like it. The smell of meat hitting that high heat, the sizzle of fat dripping down, and that crucial moment, the perfect, dark, crusty char forming on the edge of your steak. We actively look for that flavor. It's a fundamental part of cooking.
It is. And you're hitting on something universal there. That deep, desirable flavor, that intense brown crust, sometimes even black. It's all the direct result of chemical reactions, high heat breaking down proteins and sugars.
Right.
But what we're diving into today is the other side of that chemistry, the part that makes that char and the smoke that comes with it a source of real concern.
Okay.
We're going to be unpacking the science of compounds called heterocyclic amines, or HCAs, specifically PhIP, MeIQ, and MeIQx, alongside another group called Polycyclic Aromatic Hydrocarbons, or PAHs.
Those are definitely some intimidating chemical names, and they're at the very center of this deep dive. We've pulled together a stack of sources today, focusing heavily on background documents from the National Toxicology Program, some extensive epidemiological research too.
A lot to get through.
A lot. And our mission here is to understand not just what these chemicals are, but exactly how much exposure we might be getting from common foods and what the science says about the biological process that makes them, well, potential carcinogens.
Exactly. The focus is on the mechanism. See, the formation of HCAs is a natural outcome of cooking at high temperatures, but it requires specific biological components to be there in the first place.
What kind of components?
HCAs are formed when creatinine, which is super concentrated in muscle tissue and amino acids, the building blocks of protein, condense together. It's essentially a heat-driven fusion reaction.
So if you have muscle tissue and you crank up the heat?
You have the recipe for HCAs, precisely.
Yeah.
And the higher the temperature and the longer you cook it, the more of these compounds you're going to make.
Okay, what about the other group? The PAHs.
The PAH have a different but related origin. They form when fat and juices drip out of the meat onto a really hot surface like charcoal or even your gas burners.
The flare-up.
Exactly, the flare-ups. That fat basically pyrolyzes, it creates smoke, and then that smoke rises and coats the outside of the meat with PAHs. So you get this double whammy of chemical formation, all driven by aggressive heat.
Okay, let's unpack the chemistry of the HCAs themselves.
Sure.
We're focusing on what you called the big three. Chemically, they're amino-imidazolines. What's so special about their structure?
Well, structurally, they all share a common foundation, an imidazole ring. But what makes them so potent is their shape. Now, here's a cool chemistry detail for you. MeIQ and MeIQx, they're what we call fully planar aromatic structures.
Planar, like completely flat.
Exactly. Imagine them like rigid, flat little playing cards.
Okay.
But PhIP, which stands for a much longer name, 2-amino-1-methyl-6-phenylimidazo-4,5-bipyridine.
Right, PhIP is easier.
PhIP is different. It has an extra group, a phenyl moiety, that is not necessarily coplanar with the main ring. So you can imagine a piece of it sticking up or twisted just a little bit off that flat plane.
And that little twist, that change in shape, I'm guessing that's significant.
It's very significant for how the molecule interacts with biological structures later on. That slight twist can change everything about how it fits into a cellular pathway.
And just to ground this in reality, these are purely a product of cooking, right? You can't just go buy these chemicals.
No, not at all. They have zero commercial uses. The only reason they exist in a lab is because researchers synthesize them specifically to study their toxicity. Our entire exposure comes from the food we cook and eat.
So we're finding them in cooked beef, pork, chicken, fish.
Consistently, yes.
Let's get to the data then. If PhIP is the chemically weird one, is it also the most common one we're exposed to? And how big is the difference between, say, a rare steak and one that's well done?
The data points are really stark. And yes, PhIP is generally the most abundant HCA detected in cooked meats, and that high heat spike you mentioned, it's exponential.
Really?
The sources show data from fried beef patties when they were cooked at a moderate 150 degrees Celsius, that's about 302 Fahrenheit, for 4 minutes. PhIP was not detected. Zero.
Completely undetectable at moderate heat.
Gone. But when they ramped that temperature way up to 230 Celsius or 446 Fahrenheit and cooked it for 10 minutes, Now you're talking well done with a good crust.
Mm-hmm.
The PhIP levels just surged. They shot up to 32 nanograms per gram. So you're seeing a direct proportional relationship between temperature, time, and the creation of these compounds.
It's like you cross a temperature threshold and the reaction just takes off. And this brings us to what I think was the biggest surprise in all the source material.
I think I know where you're going.
The chicken data.
This is the definitive aha moment. It completely flips our assumptions about what's safe on the grill. Very well done grilled or barbecued skinless, boneless chicken breasts, the thing everyone thinks is the healthy, lean choice.
The healthy option, yeah.
Produced the absolute highest levels of PhIP detected in the data. An astonishing 480 nanograms per gram.
480? That's more than 14 times the amount you just said was in the well-done hamburger.
Exactly. The hamburgers clocked in around 32.8 nanograms per gram. And the reason for the difference, it highlights that HCA formation is less about the type of animal and more about surface chemistry.
How so?
A chicken breast is lean and thin. That means the muscle tissue is rapidly exposed to very high, very dry heat. You create a huge amount of surface char relative to the mass of the meat. If you cook that to a crispy charred state, you are absolutely maximizing HCA production.
So the key takeaway here is that the cooking method and how done you make it are way more important than what animal it came from.
That's the critical point, but we also have to talk about where else these things are hiding.
Right, because it's not just in the meat itself.
No. The hidden risk is in anything that comes into contact with the meat juice and the high heat. We're talking about the pan scrapings, the fat drippings. These are HCA super concentrators. The sources showed pan scrapings with up to 144 nanograms per gram of PHE.
So if you're making a pan sauce, deglazing with wine or stock, you're not just getting flavor. You are actively concentrating the HCAs and then pouring them right back over your food.
Precisely. And it doesn't even stop there. PhIP and MeIQx have been detected in the aerosol droplets formed during cooking.
You mean the smoke?
The smoke, the steam, yes. So the person standing over the grill inhaling that amazing smell is also getting a low level of potential inhalation exposure.
Okay, so the exposure routes are clear and potent. Let's move to section 2, the regulatory and biological context. I want to start with the big picture, the IRC classifications that really kicked off a lot of this research.
This is a crucial context bridge, yeah. The International Agency for Research on Cancer classified processed meat, so think bacon, ham, hot dogs, as carcinogenic to humans, group 1.
Group 1, that's the highest level, right? With things like asbestos and tobacco smoke.
That's the one, based on sufficient evidence linking it directly to colorectal cancer.
And what about regular unprocessed red meat, like a steak or a pork chop?
That was classified one step lower. Unprocessed red meat was placed in Group 2A, probably carcinogenic to humans. And that probably means the evidence is limited in humans but sufficient in experimental animals. It was that huge decision that really pushed researchers to pin down the specific chemical culprits, and HCAs were prime suspects from the start.
Okay, so if HCAs are the mechanistic link, how dangerous are they on their own? You said they're potent mutagens?
Highly potent. In standard genotoxicity tests, like the Salmonella assay, which measures a substance's ability to cause mutations, MeIQ and MeIQx were found to be more potent than aflatoxin B1.
Aflatoxin, that's the mold on peanuts, the famous liver toxin.
Yeah.
The very one. And crucially, all 3 of the HCAs we're discussing are significantly more potent as mutagens than benzoylpyrene, which is the classic PAH carcinogen we always associate with smoke.
Wow. So molecule for molecule, they're more efficient at scrambling DNA, but our bodies don't just absorb them as is, right? There's a process.
That's the key distinction. HCAs are what we call procarcinogens.
Yeah.
Think of them as chemical sleeper agents. They're relatively harmless until our liver decides to activate them.
And that activation happens in a 2-step process, you said.
A mandatory 2-step metabolic process, yes.
Okay, so let's break that down. Step 1.
Step 1 is phase I activation. It's like the first wake-up call. It happens mainly in the liver using an enzyme called CYP1A2. All this step does is add a hydroxyl group to the HCA molecule. This makes it more water-soluble, so the body can, in theory, get rid of it.
What?
But this new intermediate molecule is now chemically primed for damage. The body's attempt to detoxify it actually sets the stage for the danger.
I see. So it's waiting for step 2. It's waiting for phase 2 activation. This is where other enzymes like N-acetyltransferase or NAT come in. These enzymes modify that intermediate again, and they create the final incredibly dangerous molecule, the aryl nitrenium ion.
The aryl nitrenium ion. That's the ultimate villain here. What does it do?
It's extremely reactive, it's desperate to bind to something, and its favorite target is DNA. When it binds, it forms what we call a DNA adduct.
An adduct.
Imagine trying to read a line of code, but someone has glued a big, clunky chemical block right in the middle of a letter. The code is now unreadable, or worse, it reads as something else entirely.
It's a mutation, the necessary first step for cancer to develop. So what do the animal studies tell us about where these HCAs attack? Is it just the gut?
No, it's a diverse systemic effect. PhIP, for instance, it caused lymphomas in mice, but in rats, it caused tumors in the colon, the mammary gland, and the prostate.
That's all over the body.
It seems to affect a wide variety of tissues. PIQ caused tumors in the oral cavity, the stomach, the liver. They're not one-trick ponies.
You also noted an interesting counterexample in the sources with MeIQx.
Yes, so MeIQx caused liver and lung tumors in mice, but in a very long 84-month study in cynomolgus monkeys, it was not found to be carcinogenic.
Why not?
The thinking is that that particular species has a very low level of metabolic activation. Their bodies just aren't very good at running that two-step activation process we just talked about.
Which brings us to the most important question. What about humans? Are we good at it?
The sources confirm we are. We are active metabolizers. HCAs form DNA adducts in human tissues, even at the kind of doses you'd get from a normal diet. And what's really compelling is the comparative data. Human colon tissues sometimes shows higher levels of PhIP and MeIQx DNA adducts than you'd find in rats, given equivalent doses.
So we seem to be quite effective at turning these things on.
Very effective.
This is the crux of the human exposure dilemma then. We know these compounds are potent, we know we're eating them, and we know our bodies are efficiently activating them into DNA-damaging agents. So why is it so hard for epidemiology to draw a clean, definitive line between specific HCAs and human cancer risk?
It's the challenge of what we call confounding variables. HCAs are never, ever consumed in isolation. They're highly correlated with each other. They're embedded in a complex matrix of fat, protein, and all those PAHs we mentioned at the start. It's like trying to figure out the effect of one specific grain of salt when you're eating an entire salted meal. It's nearly impossible to isolate one factor.
That makes a lot of sense. But the sources did give us two pretty strong pieces of evidence that connect the dots.
Absolutely. First, there's the direct dietary evidence. A study showed vegetarians had significantly lower levels of PhIP bound to proteins in their blood compared to meat eaters.
Clear confirmation of the source.
No question. And second, there's the genetic susceptibility factor, which comes right back to that metabolic activation pathway.
Tell us more about these rapid acetylators.
Okay, remember that key phase 2 activation enzyme, NAT? Well, there's genetic variability in humans in how fast that enzyme works. Some people are classified as rapid acetylators, others are slow acetylators.
And the rapid ones would be at higher risk.
That's the theory. Because they have a more efficient NAT enzyme, they might be generating that final, highly reactive aryl nitronium ion more quickly, more effectively. That could lead to more DNA adducts in their tissues compared to a slow acetylator eating the exact same charred burger.
So your own body's genetic machinery plays a huge role in your personal risk. That's a profound finding. And it leads us perfectly to the most actionable part of this whole dive. If the risk is all about heat and time, how can we still enjoy grilling without maximizing our HCA exposure?
Thankfully, the science on mitigation is pretty clear, and the solutions are straightforward. It all comes back to managing temperature and surface area.
Okay, let's start with changes you can make right at the grill.
Number 1, and this is the simplest, cook longer at a lower temperature, avoid that peak heat window, and you avoid that exponential surge in HCA formation we saw in the beef patty data.
And for the PAHs?
For PAHs, avoid direct flame exposure. Have a spray bottle with water handy. When fat drips and causes a flare-up, douse it. That smoke is what's depositing PAHs on your meat.
Minimize the burning, minimize the smoke, minimize the PAHs.
Got it.
What about prep work before anything even hits the heat?
Prep makes a huge difference. First, you can trim the visible fat off the meat. That removes the fuel for those PAH-generating flare-ups. And second, marinate the meat.
Does marinating really work?
It does, especially with acidic or antioxidant-rich marinades, things with herbs and spices. It creates a physical barrier that can reduce HCA formation.
That's a great example of science and flavor working together. Right. And there was a simple kitchen hack involving the microwave, too.
Yes, partially pre-cook the meat in a microwave before you put it on the grill. This cuts down the time the meat needs on that high, dry heat, which cuts down the window for HCA creation.
And when it's actually cooking?
You should turn the meat frequently. Just continuously turning it over helps keep the surface temperature more moderate and reduces HCA formation. And choose wisely. Thinner cuts or seafood cook much faster, less time on the heat, less HCA formation.
And I guess the easiest way to improve the balance of what's on your plate
Is to load it up with produce. Fruits and vegetables do not form HCAs. It's a guaranteed way to lower the overall HCA intake of your meal while still enjoying that grilled flavor.
So to wrap this all up for you at home, what does this mean for your next cookout? We've seen that these heterocyclic amines, MeIQ, MeIQx, are potent mutagens. They're in the same chemical potency range as some really well-known toxins. They're formed by high heat in muscle tissue, especially when it's charred or very well done. And critically, the human body is very efficient at metabolizing them into DNA-damaging agents.
And we know that the formation of those DNA adducts is considered a necessary precursor for carcinogenesis. We also know that the efficiency of that critical metabolic activation pathway is directly influenced by your individual genetic factors, like being a rapid or slow acetylator.
Which leaves us with this final provocative thought for you to take away. If your unique genetic makeup dictates how efficiently your body turns these common dietary compounds into DNA-binding carcinogens, does that mean the health risk of grilling is less about generalized population statistics and much more about knowing your own individual metabolic chemistry? That's something to mull over the next time you have to decide just how dark you want that crust to be.